School of Integrative Plant Sciences, New York, USA. He also serves on executive board of Cornell Institute for Digital Agriculture. He has over 20 years of experience in plant genetics, plant breeding, biodiversity, international agricultural development, and capacity building across 5 continents involving 6 crops. Previously, he led a global research program as a senior scientist at International Potato Center in Lima, Peru, with several projects in Central and South Asia, Sub-Saharan African, and Latin America. He has extensive experience in mentoring graduate students, training postdocs, scientists, and institutional and intellectual capacity building in both global north and global south. His MSc is from University of Göttingen in Germany and PhD is from Zurich in Switzerland. He then completed postdoctoral research at University of York in UK and University of Illinois Urbana in US. In his scientific career, he has secured funding from various prestigious sources, including Bill and Melinda Gates Foundation, USDA, AFRI, AFRI, NSF US, NSF China, and Horizon 2020. To name, and these are just few of those. He has published over 100 articles in high-impact scientific journals, including Science, Nature Genetics, and Genome Research. His research has been highlighted in popular magazines, including Science Magazine, Economist, New York Times, and Forbes. So we welcome you at camp, Dr. Sir. Please come on, guys, and give your talk on genetic improvement of disease resistance in delicious apples. Please give a big round of applause to Dr. Awais Khan.
Thank you. Okay, so thank you very much. Can you hear me? I have been traveling for last 40 hours. So and I really didn't sleep very well last night. And if I don't say, or if I say something which is not okay, just excuse me, okay? So yeah, thank you very much for the introduction, and thank you very much for for hosting me, and also thank you very much for listening me, because it's really wonderful to be here at CAM. When I was a BSc honors student in agriculture college in Kashmir. I'm originally from Azad Kashmir. I was always imagining and dreaming to be at camp. So I'm really happy to be here today. So I think I was saying to Dr. Tayyib that I might have been here like maybe 26 years ago, but as a young student and trying to see if I can get, somebody can accept me here. But yeah, it didn't happen. Okay, so I will be talking to you about apples, but I think apples as an example, but I will be talking more about how to utilize crop genetic resources to breed disease-resistant fruit crops. And I think you can also utilize this for any other crop system. But I think most important thing usually I have seen from my talks when I travel internationally, that young people and students, they ask me questions about my career and also the graduate and scientific life in the US and in Europe. So feel free to stop me and ask me those questions, or maybe at the end we can have discussion about that. Okay? So let me see. Sorry. Which? I think here. You point towards this one. Okay. अभी point. इसकी तरफ ये पॉइंट करके ना
लगा के देख लेते हैं यार। सर वर्क नहीं कर रहा, इससे गुजारा हो जाएगा सर?
Okay, so I think the pointer is not working, so I will try to change from here. Okay, that's fine. Oh, I think everything is. Even my laptop is not working. Maybe escape and we try again. This one? Okay. Okay, sorry for the glitch. So, as I said, I'm Awais Khan and I'm originally from Azad Kashmir. I did my undergraduate bachelor degree from University College of Agriculture in Rawalakot, but now it's Punch University and there's agriculture faculty in there. After that, I went to Göttingen, Germany to do my master's, and I did my master's in plant breeding and genetics from University of Göttingen. And then I went to Switzerland where I did my PhD, and this is ETH Zurich. I did my PhD on genetics of disease resistance, fire blight resistance, a bacterial disease of apples in Switzerland. And then I thought, okay, let's move towards more west. So I went to UK to do my postdoc, and I did my postdoc from University of York in UK, and that was on genetic diversity and genetic mapping of artemisinin-related compounds in Artemisia annua, a medicinal plant. And this artemisinin, this terpenoid, is very important and used in medicinal, used in drugs which are anti-malarial drugs. So I can tell you a little bit about link of this Artemisia project and my Rawalakot experience. So my graduate supervisor was Dr. Dilnawaz Ahmad Gardezi, and some of you know him, and he gave us training to work on genetic diversity of medicinal plants in Rawalakot, and we didn't have many equipments and many systems, but he really cultivated deep interest in us about medicinal plants. So when I did my PhD, I saw a position in UK to work on medicinal plant, and I left everything and I said, okay, I want to go and work on medicinal plants. So I worked 2 years on this medicinal plant Artemisia annua in UK. And then I went to University of Illinois Urbana-Champaign in 2010 to work on genetics of disease resistance in apples and also pear, and that was on genomics and genetics of apples and pear. And in 2012, I decided to take a position in Peru, South America, and in Peru, it was a global lead for abiotic stress in potatoes and sweet potatoes. And there I was working in Lima, Peru, but my position was responsible for abiotic stress tolerance in globally. Basically, I was working in sub-Saharan Africa, I was working in India, I was working in Bangladesh, I was working in Central Asia. I traveled to Malawi, Uganda, Tanzania, Kenya, Ethiopia. I mean, you name these countries, and we were selecting heat, drought, and salt-tolerant potatoes. And I was more on the genetics of abiotic stress tolerance. So I think maybe I can pause here and tell you that how I decided to go. I think many students, they ask me this question, how I decided to go to Germany in 2001 from Rawalakot. So I finished, I think, in July, and October 2001, I was in Germany. And um, basically, I'm from a very poor village background. And we used to, I mean, we didn't have much. Our house used to be mud and clay, and we didn't have really, I mean, animals were also in the same on one side of the house and so on. So you can imagine that kind of environment I was growing in. And also going to the when I was thinking to write these letters. Can you hear me? Yeah. When I was thinking to get admission somewhere in Europe, I didn't also have uh, much geographical understanding of Europe. I was thinking, okay, I want to go to West because I will have better future options there and better career and education. And I didn't know how to use computer. I went to Rawalpindi and I gave, I think, $50 to somebody to create my email account, and they created Awais Khan PK 2000, like a flight number or something, 2000 at yahoo.com. So that was my computer knowledge. And um, so I knew one guy who could write, who could type in computer. So I gave some, actually, I wrote a letter with hand, gave to him, and he typed that on computer, and he gave me one copy, and it was written to the embassy of dash dash dash. I am this student. And then I made many copies, and then I filled which embassy, and I asked question about if in your country, if there is any agriculture education, and this is my background, and could you please provide me more information. So that was kind of the approach I tried at that time. So I didn't know which countries have embassies in those in Islamabad. So I sent those letters in Islamabad, and some embassies which existed, they responded back to me, and some didn't respond it. So that was the screening. And then I got those letters and a lot of pamphlets, information, and then I started to look which universities I should be applying. And then criteria was which university is cheap. And one day my father, he used to listen radio, and my mom used to fight with him, saying that you always listen this radio and nothing comes out of it. And he said he used to listen this voice of Tehran, voice of Deutsche Welle, Urdu service. So he told my mom, no, he told me that, Awais, I heard today that there is a Pakistani student in Germany and saying that education is free in Germany. So why don't you apply to Germany? So that's how I applied to Germany. And also we didn't have much money, and my father was very reputable in the village. He had a bookshop, and he was helping other kids for education. So he asked people in the village, some relatives, and he borrowed money, and he gave me money for ticket and stuff. So that's how I ended up in Germany. But when I got to Germany, I realized, and my father also told me, that you should focus on education. Don't try to get in the trap of making money, because I have 5 sisters, and our house and economic situation I told you. So I could have been been focusing on helping the family and making money. But somehow my father and my sisters told me that I should focus on education. So that's my story. That in Göttingen, I had a very nice professor in plant breeding genetics, and he taught me about plant breeding genetics, and that's how I got into this area. So anyhow, you can ask me more questions later, or by email, or even after the presentation. So that's a different topic. Okay, now I get back to my science. Okay. So in 2012, I was in International Potato Center, and then I decided to join Cornell University as an associate professor to work on apple breeding and genetics. And Cornell has different campuses. The main campus of Cornell University is in Ithaca, and there's an agriculture campus which is called Cornell Agritech. And Cornell is in New York City. Oh, sorry, New York State. New York State is very big. And when I tell people that I am in New York State, they think I am in New York City. But actually, we are very close to Toronto, Niagara Falls. We are 5 hours far from New York City. So Cornell is the only university which is public and private university. It has public mission, but it's also private. It's Ivy League University. It's one of the top 8 universities in the US. And the region where this both Cornell main campus and Cornell Agritech is called Finger Lakes area. We have 5 lakes there, and also Lake Ontario. It's a very beautiful area, and especially in summer and fall, it's beautiful area to come and visit. Niagara Falls is very close to us. So one of the reason for me to join Cornell was Cornell has historically very strong apple genetics and breeding program. The oldest breeding program in the US, the apple breeding program, is at Cornell. Cornell has the world's largest apple collection. So I was always inspired by genetic diversity. So Cornell had more than 5,000 different accessions of apples, almost representing all species of malus. So that was one of the main attraction for me. Cornell has also the apple rootstock breeding program, and Cornell has released many successful apple sign varieties and also rootstock varieties. And also, I think many of you know about gene gun. So gene gun was also invented at Cornell. There are many Nobel laureates from Cornell, more than 50 Nobel laureates. So it's a very good university to be at. So I'm very lucky. Now you see my travel from Rawalakot and from that to one of the best universities in the US. So my program at Cornell, when I joined it, I established this program to work on the providing sustainable disease management options for apple orchards or apple industry. And basically, it includes the main focus is genetics of fire blight and apple scab resistance. These are 2 major diseases of apples. One disease is called fire blight. Fire blight is a bacterial disease of apples, and apple scab is a fungal disease of apples. And they are present most parts of the world where the apples are grown. So one part of my research program is the quantitative genetics. So I work on genetic mapping of disease resistance. So I use QTL mapping approach, biparental QTL mapping. We make cross, generate mapping populations and do the QTL mapping for the disease resistance. But we also use genetic diversity, GWAS panels to identify genetic basis of disease resistance. Then another fascination I have from my potato and sweet potato time is to work on root system architecture of apples. So potatoes and sweet potatoes, they are underground. So I used to work with underground tubers and roots, storage roots. So I developed that fascination of studying root system, and that's how I'm still working on root system of apples. Then also at the same time, we look at genetic diversity of pathogens, because you have to know about the pathogens to really identify appropriate genetic resistance for those pathogens, because you can have different pathogen strains, they can have different effectors, and if you identify a QTL by genetic mapping, either QTL biparental mapping or GWAS, and maybe it is not effective against wide collection of strands, and it's very specific to one strand, so then it's not very useful. So using this information, I try to develop DNA markers, and these DNA markers are supposed to be used in breeding programs to develop disease-resistant cultivars. We also do functional validation of these QTLs and the genes underlying these QTLs using genome editing and transformation to see if these are really actual candidate genes, actual genes behind that. So I'm not looking at my next slide, so I don't know what's if I'm already telling you about this. But one thing, why to work with apples and why to work with vegetables and the fruits. Malnutrition, and also people work in biofortification and nutrition. So malnutrition is still a major problem in the world. Sometimes we eat nice meal and we think people are well fed, but even having enough food to eat doesn't mean that we are eating nutritious food. So um, more than 2 billion people in the world, they are still malnourished. They don't have essential nutrients requirements. So and that's not only in Africa or South Asia. It's also a global problem. In Europe and America, you also see people which are deficient in these nutrients. So working on vegetables and fruits, that's very rewarding because you are really trying to fulfill the requirements of the nutritions. And for the proper functioning of the society, nutrition is very important. I mean, food security is important, but the nutritional security is even more important. So now in this image, I'm showing you that if, let's say, if you walk to an apple orchard, what will you see? You will see an apple orchard without, if you don't manage it, you will see all these kind of diseases in an apple orchard. You will see apple scab, you will see fire blight, you will see alternaria, you will see rust, you name it. And this is, I'm just talking about the diseases. You will see the insects also. You will see so many problems. And we should be very thankful to the apple growers and the farmers who produce very clean fruit for us. But how many of you would like to eat one of these fruits? I don't think anyone will eat this fruit. So consumers, they don't eat this fruit. And then if you go to market, you will see very clean fruit. But if you have a disease, and those diseases which I showed you in the previous slide, they're everywhere. And what you also see in commercial orchards, and not only apple orchards but also other crops, you can see monocultures. You have one genetics which is on acres and acres and hectares. The whole region has only one variety, one genotype, one cultivar. And if that is susceptible to that pathogen, if a new pathogen emerges which can infect that crop, then you will have devastation. So like you see on your left side, that it can wipe out the whole orchard within days. And the apple growers, they use heavy fungicide applications or antibiotics, chemical applications. And chemical applications have a lot of impact on health and environment, and also a heavy cost. But now if you think about these issues are there, but how to improve it? And if you are trying to improve it from breeding approach, apples, the juvenile phase of apples is 5 to 7 years. So the flowering, if you grow apples from seeds, the flowering starts in 7 years or 5 years. For example, 5 years. And if you are making a cross, most of the cultivated apples are the apples we eat. They are highly susceptible. So most of the resistance is present in wild apples. So if you are crossing a wild apple with a domesticated apple, you need 5 cycles of crossing to retrieve good domesticated background, good quality, even not sure. And if each cycle takes 5 years, 5 multiplied by 5 is 25 years. And a breeder's age is also, professional age is also 25, 30 years or 35 years. Yeah. So it's really a big challenge to breed apples or long juvenile phased perennial trees. So here I am showing you these are 4 chromosomes of apples. Apples have 17 chromosomes. So these are 4 chromosomes of apples, and this resistance on chromosome 3, linkage group 3, is from this fruit. Just look at the fruit down there. And most of this fruit is not eatable. Nobody would like to eat this. And the same problem which I said previously, that if you are crossing to introgress this linkage group 3 resistance for fire blight in domesticated, you need 25-30 years. So it takes long time. So but with marker assisted selection you can speed up the process, but still it takes long time because juvenile phase will be still there. Yeah, long. So we also see this issue, and this is one of our paper we published, that the most important resistance for apple scab is called VF. I mean, you don't have to, you are working on different systems, but just believe me that the most important apple scab resistant gene is called VF, and more than 80% of the cultivars which are scab resistant, they had this resistant gene. And there are strains of pathogen which can overcome this resistance. We also identified them in New York. So what does it mean? That the resistance is broken down. So you have to look for new resistance. And it took about 30 to 40 years to breed for that resistance. So also major resistances can break down, new strains can come which can overcome this resistance. So what does it mean? We need to identify new sources of resistance. And to identify new sources of resistance, we have to understand the domestication history of that crop, that plant. So for example, here in apples, what I was telling earlier to some colleagues, that apples originate from Central Asia. So if you know about host and pathogen coevolution, some host and pathogen, they co-evolve side by side. So if you know the domestication history of apples, you know that the resistance to some of the pathogens might have been in co-evolved in Central Asia. So you have to look for the species which are in that region. Or if you want to look for resistance for that specific disease, you have to go and screen those accessions. So domestication history is very important to understand if you're looking for natural resistance. So as I was saying earlier, that we are very lucky at Cornell that we have the world's largest collection of apples. So we have this diversity which is close to 4,000-5,000 apples. So I cannot show you disease resistance in a beautiful picture or figure. This is just showing you the snapshot of this collection, showing the flowers and showing the fruit. And you can see that there's so much diversity in this collection. So what I do in my program, we do the identification of genetic sources of disease resistance. We collect these accessions, we graft them, we make replications, and then we have the pathogen strands and we infect them and we identify the most resistant accessions. So in this case, for example, we are identifying for scab resistance. And in green, you see healthy. So these accessions, we infected them and they didn't, this was in the field and they didn't get infection. So and this data was collected for 3 years. So we have very robust resistance in these accessions in the field conditions. We have very susceptible as well, side by side. So the same thing we also did for fire blight. We also grafted them in the for fire blight. We do controlled inoculation in the greenhouse as well, and then we screened and identified the most resistant accessions. And then what we do once we identify this, another thing in my research program, we are developing new ways how to speed up the genetic mapping process. So genetic mapping, normally the quantitative way of genetic mapping, what we do, we cross 2 parents, susceptible and resistant. We have F1 segregating for that resistance, and then we map. But it can take long time. And also if these are apples, it needs a lot of space. and also 4 years, 5 years, 10 years. And if it's only one resistant gene you're mapping, it's not very economic. So these are some of the constraints. So we thought, okay, let's try to develop a new way to speed up the process of mapping. So we are developing this method based on open pollinated mapping. And we in this method, what we are doing is bees are making crosses all the time. So if we know that this mother plant is resistant, there will be fruit on this mother plant naturally by natural crossing, and we can do genotyping and we can do phenotyping on the F1s from those seeds, and we know what is coming from mother, and we know about the whole orchard, and we can also deduce which pollen donor has contributed to that. So in using that information, we can also map resistance in this open pollinated. So we don't have to maintain that new population. We don't have to make the cross. So this is a new method we have been developing. So what I was showing to you, we do the screening, but we also developing new methods of genetic mapping. Also, to speed up the process, we are developing new high throughput methods of screening. Because again, imagine if we are infecting apples with fire blight. Fire blight is a disease which kills the trees. Okay, so now you have single seed grown plant. You infect with fire blight, it will kill the plant. You will not have any plant left. It has potential. So you have to make many replications, and it takes a lot of time, grafting and then growing them, and greenhouse space and everything. So what we have been doing, we have developed a method based on leaf discs. So we make small discs of leaves, and then we infect them with fire blight, and we use image analysis to quantify how much infection happened. And plants always make new leaves, so we can have many replications. And we can combine that data to have very reliable result for that accession and next accession, and then use that information for genetic mapping. So this is also a new method we developed, and we published it for phenotyping. Then we have also developed a new method of DNA marker system for apples. So most of the marker systems which were developed for apples, they were biased towards domesticated apples. So basically, if you have genome sequence of domesticated apples and you develop DNA markers based on that sequence, then it will be biased towards that domesticated sequence, domesticated apples. But I told you that domesticated apples are susceptible to diseases. So now what I'm saying is you are mapping it from the wild accessions, wild apples. So if your marker system is biased towards domesticated apples, you cannot map. There will be missing data for your wild apples. So we have developed a method based on pangenomes, where we have genome sequence of many species, and we found the core genome which is conserved across many species. So we went for 14 genomes, and then we saw where we have the best coverage, and about 12 genomes we have best coverage, and we use that coverage to develop markers. Now these marker systems based on DART in Australia, a company, DART tag. So we have close to 6,000 markers which can work on most of these wild species. So we can use this marker system for genetic mapping from wild species. And we have shown this for fruit weight, fruit length, and different traits, for association mapping, and also for QTL mapping, and also for pedigree analysis. So this marker system works very nicely. And then we are using these marker systems and phenotyping method I showed you for mapping. So here, for example, I'm showing you that we have mapped for fire blight resistance from Gala and crossed with 2 Kazakhstan apples, because again Kazakhstan is the center of origin of domesticated apples. So resistance for many diseases comes from the apples which developed there for generations. So we mapped one on chromosome 7 and another. Actually, here in the example, you're seeing the genetic mapping of fire blood resistance on chromosome 7. So here you see chromosome 7 and the peak is there for this QTL. So this was biparental QTL mapping, but also we are doing fine mapping of the QTLs which have been identified by other groups previously or our own group previously. So QTL region, I don't know how many of you are working with QTLs, but you work mostly with the targeted genes in CEMB. But QTL region is usually a big region, centimorgan, maybe 5 centimorgan long, or even 10 centimorgan, could be hundreds of genes or 10 candidate genes. So then you have to narrow it down so that you know which are the most probable candidate genes. And to do that, it's not very easy process. People used to do chromosome walking and so on. But in this case, what we are fine mapping in this example, there are 3 resistance loci on chromosome 1 of apples for apple scab. One is from Antonovka apples, another is from Honeycrisp, and third one is which have been compromised. I showed you in previous example, it was broken down, VF. Okay, so they are all on chromosome 1 and they are very close to each other. But I wanted to separate them, that which are they same, are they different? And we have sequenced these 3 genomes, and we have developed haplome level or phased assembly for these three cultivars, three genomes. And now we have narrowed down. Then we develop the populations, we develop the markers, and we identify the recombinants, and we narrow down this region to very few candidate genes. And now we are functionally validating those candidate genes. I think I have gone through. So for functionally validating these candidate genes, we are using genome editing and also using transformation. I mean, still it's in early phases, but we are also, I mean, tissue culture is very specific to different cultivars, and we have been struggling a lot with the tissue culture of Honeycrisp and Antonovka, but we are doing this both gain of the function and loss of the function in some of the examples here. But I think the most exciting thing which I wanted to show you here is this one, because these are the previous examples I showed you: screening and mapping and fine mapping. But in this example, and functional validation. But in this example, I want to show you that we are also developing pre-breeding lines by speeding up the process. So these resistant genes which have been identified previously are by our own group. We are trying to make new lines by combining multiple resistant genes in one background. And as I told you, in apples, if juvenile phase is 5 years, it can take 25 years to bring those genes together. Yeah, and PhD students, they will be working only for 5 years in the lab. So how to do that? So we are using this transgenic approach, early flowering transgenic approach. There's MADS box gene called PP-MADS4. Yeah, sure.
Is there any relationship this MAD with FLC? There's also the gene that actually controls the vegetative growth. Whether there is any interaction between these cells?
I don't know specifically about that, but it's in the same group like FT.
And FT actually activated after that, when there is flower initiation. Constant genes that activate. This is another group. This is FLC that is controlled in apple. If you say that FLC vernalization actually control that gene.
Okay, so I think we can look more into that. I don't know specifically on that. Yeah. So I think again it's a fruitful homologue. like in Arabidopsis you have fruitful, and it's very similar to that gene, and it is very good for this transition from vegetative growth to reproductive stage. And this gene has been used in apples by a group in Germany from JKI to show in apples that if you put this gene in apples, it's actually coming from silver birch, and this gene, if you have in apples, instead of 5 years, you can have apples flowering in 6 months or 7 months. I mean, it can be different for different genetic backgrounds, but we have seen from 4 months to 8 months flowering. So now you can imagine how drastically it changes that the generation cycle. And we have been using this to speed up the process of combining the genes in one background. I mean, the transgene, we have the transgenic line in the greenhouse, and we use that transgenic line and cross it with a donor accession which has resistant gene. And donor accession is usually the wild crab apples. And then first cross, we select for the resistant gene and also early flowering. And then we have both in same background, and then we combine different combinations, and in that way we can combine multiple genes in a very quick fashion. And we are also characterizing S genes because apples have S alleles and they cannot combine always cross with each other. So we have to change the recurrent parents. And we have now lines with 2 genes and 3 genes in combination, and we are working actually here in the next slide. You see, we are working with 3 diseases: fire blight, apple scab, powdery mildew, and for each disease we have 3 to 4 genes or QTLs, and we are using flanking markers to do the selection. And this is working very nicely, and we are aiming to have 2 or 3 genes in combination. And the idea is that we have pre-breeding lines available, and those could be used by the breeders to develop the final good fruit quality and disease-resistant apples. So with that, I think this is one of my final slide, that what I tried to show you, that if you would like to use genetic diversity, genomic germplasm, this is the efficiency, the way I look at it, that you start with the genetic diversity, there is a linkage drag, and you can overcome this linkage drag by using markers or by using this background selection of the undesired genome using the genome sequence or pangenomes. And in that, and also if you combine it with genome editing or transformation for functional validation of some of the genes you identify, and also like early flowering, innovative kind of approaches, you can speed up the process and you can lead it to cultivars which could be nutritious and disease resistant. Otherwise, no breeder is going to make the cross with wild apples, because wild apples have linkage drag, and consumers like you and me, we want to have good fruit quality apples, not disease resistant apples. If they don't taste good, we don't eat. We don't care that they are disease resistant. So important is fruit quality, but to have the good fruit quality and disease resistance, that's very sustainable, and I think that's how we can achieve it. So with that, I have a big team who is behind all this work, and I have presented their work, not my work, and the donors who funded this work. So I didn't go in detail in any of this research, but you can ask me questions about that. So thank you.
Thank you so much, Dr. for such a nice and informative presentation. Now the house is open for general discussion, especially for questions and answers from our dear students. Please do ask. Dr. is here.
Yeah, you can ask me questions about science or Anything else?
So, yes, please.
میں لبرٹی لے کے اردو میں کوشچن پوچھنا چاہوں گا.
پنجابی پوچھو.
تھینک یو سر, بہت شکریہ آپ کے, بہت زبردست لیکچر ہے. اچھا سر, ہم جب ایوولوشن کی بات دیکھتے ہیں کہ ایک بڑا کامن سا ٹرینڈ پایا جاتا ہے, ایپل کی بات کر لیں یا پوٹیٹو میں, کہ جیسے جیسے ڈومیسٹیکیشن ہوتی ہے, کراپ ایوولوشن کی طرف بڑھتی ہے اور کلٹیویٹڈ فارمز آتی ہیں, تو ان میں سے جو ریزسٹنس کے ایلینز ہوتے ہیں وہ ہم دیکھتے ہیں کہ وہ ود دا پیسج آف ٹائم وہ ڈومیسٹیکیٹڈ اور کلٹیویٹڈ اسپیشیز سے نکل جاتے ہیں, اور پھر ہمیں سورس پھر وائلڈ سے ڈھونڈنا پڑتا ہے. اور اگر آپ اس کراپ ایپل پہ کام کر رہے ہو تو جیسے آپ بتا رہے تھے کہ 25 ایئرز لگتے ہیں, تو اٹس ریئلی چیلنجنگ فار اے بریڈر. تو ایک تو کوشچن میں سر, کوشچن نہیں, ایک آپ کا انسائٹ ہے کہ یہ ایسا کیوں ہوتا ہے, ایک ٹرینڈ ہے ڈفرینٹ کراپس میں کہ ود ایوولوشن ود دا پیسج آف ٹائم وہ جو ریزسٹنس ایلیلز ہوتے ہیں وہ ایک طرح سے ڈیلیٹ ہوتے جاتے ہیں یا نہیں ہوتے ڈیزیزز کے اگینسٹ. دوسرا اگر آپ اس پہ کچھ کمنٹ کر سکیں.
پتہ نہیں, یہ کافی بڑا کوشچن ہے, لیکن کافی اس کے آنسرز اور اسپیکٹس ہو سکتے ہیں. لیکن ایک تو یہ ہے کہ جو کوالٹی ہے, کنزیومر کوالٹی, یا جو جیسے فروٹ کوالٹی ہے, تو وہ ایک ٹریٹ ہے جس کے لیے سلیکشن جس کی فیور میں ہوتی ہے. جو باقی ٹریٹس ہے اس کے لیے اتنا زیادہ فوکس نہیں ہوتا. پھر ایپلز میں یہ ہے کہ ایپل کلونلی پروپیگیٹڈ ہیں, تو اس میں وہ جو نیچرل ایوولوشن والی بات ہے جیسے باقی کراپس میں ہے, وہ ایپلز میں اس طرح سے نہیں ہے. جیسے آپ دیکھیں کہ ایپلز کی جو جینیٹکس ہے وہ سینٹرل ایشیا کی ہے, وہاں سے وہ ٹرانسپورٹ ہوئے سلک روڈ پہ ایسٹ اور ویسٹ دونوں میں, اور یورپ میں گئے, اور یورپ میں سے 1600s میں یورپینز جب نارتھ امریکہ میں گئے, نارتھ امریکن کنٹیننٹ میں, اور وہ ایسٹ کوسٹ میں گئے, تو وہ جو سینٹرل ایشین جینیٹکس تھی جو یورپ میں بھی کچھ حد تک ایوالو ہوئی, لیکن وہاں پہ بھی سلیکشن کافی تھی, اس کو اٹھا کے اسی طرح وہ لے کے گئے کہاں, نارتھ امریکہ میں. ابھی نارتھ امریکہ میں وہ ایوالو نہیں ہوا, کیونکہ اس کے کیا ہوا, لوگوں نے وہ جو بھی کلٹیور تھا اس کو کلونلی پروپیگیٹ کرنا شروع کیا, تو دیر از نو ایوولوشن ان دیئر. تو وہ جو وہاں کے پیتھوجن ہیں نارتھ امریکہ کے, ان کے ساتھ تو ایپل کبھی بھی ایوالو نہیں ہوا. تو بہت سی کراپ کی یہ اس طرح کی کہانی ہے کہ وہ ایک جگہ سے اٹھا کے ہم لوگوں انسانوں نے دوسری جگہ پہ یکدم وہ, تو ان کو وہ ٹائم نہیں ملا ایوالو ہونے کا, وہ پیتھوجنس جو لوکل تھے. تو ابھی جو نارتھ امریکہ میں جتنے بھی ایپل کلٹیویٹ ہوتے ہیں, اور وہی پھر پوری دنیا میں وہی پھیلے ہیں ایپلس, واپس بھی آئے ہیں ادھر سے ہی, صحیح. تو وہ جو ایپلز ہیں وہ گیلا ہے ایپل, تو وہ کلونلی پروپیگیٹ ہو رہا ہے, وہ کبھی بھی وہ نیچرل سلیکشن یا ایوولوشن پروسیس میں گیا ہی نہیں ہے نارتھ امریکہ میں. پتہ نہیں اور بھی اسپیکٹ ہوں گے. ڈاکٹر صاحب آپ, طیب صاحب کوئی.
ڈاکٹر صاحب تھینک یو ویری مچ. بڑا سٹڈی یہ جو ٹاپک ہے اس حساب نے بتایا, سٹڈی سے اس کا پروسیس کافی کم ہو جاتا ہے. ایز اے پلانٹ بریڈر بیسیکلی محمد ارشد جاوید میرا نام ہے, تو
صحیح.
پلانٹ بریڈر بیسیکلی جب بھی کوالٹی کی بات ہوتی ہے سر, تو ریزسٹنس ہمیشہ کم ہو جاتی ہے. یہ نیچرل پروسیس ہے. جب کوالٹی کا چیز آتے آتے ہیں جس طرح کی چیز کے اندر اسی طرح سے اس کی ریزسٹنس آٹومیٹکلی وہاں پہ ڈکریز ہوگی. نمبر ون. نمبر ٹو, جتنا بھی کسی کو پیمپرڈ کر دیں گے نا جی, اس کی ریزسٹنس کم ہو جاتی ہے. جب ڈومیسٹیکیشن کی بات کریں وہ پیمپرڈ ہوگا نہیں جی, اور جب یہ پیمپرڈ کرے گی تو اس کی ریزسٹنس کم ہو جائے گی. تھینک یو سر.
صحیح.
اچھا سر, میرا کوشچن یہ ہے کہ جیسے ہم پلانٹ بریڈنگ میں ڈیزیز ریزسٹنس کے لیے بیک کراس بریڈنگ یوز کرتے ہیں. تو سر ایپلز میں بھی کون سا میتھڈ ہے یہ سر کانگریس کا گائیڈ لائن جی کیونکہ ہم ڈیزیز ریزسٹنس کے لیے کوئی بھی اس کے لیے بیک کراس بریڈنگ ہے, تو یہ کیونکہ بگر پلانٹس ہیں تو ان کے لیے سر کون سا میتھڈ آپ یوز کر رہے ہیں؟
ایپلس میں بھی آپ یہ ہے کہ آپ ریکرنٹ جو پیرنٹ ہے وہ آپ کو چینج کرنا ہوگا, کیونکہ ایس الیلز ہیں, آپ یعنی سیلف انکمپیٹیبلٹی الیلز ہے. تو نارملی لوگوں نے جو ہے نا وہ بنائے ہوتے ہیں کہ میرے پاس یہ الیلز ہیں. تو اگر آپ جیسے ایک گیلا ایپلز ہیں تو اس کی آپ نے جو اس کا دوسرا پیرنٹ ہے اس کے ساتھ آپ نے کراس کیا ہے, تو پھر آپ کو پیرنٹ دوسرے چینج کرنے ہوں تاکہ ایس الیل ڈائیورسیفائی ہو. تو کیا کہتے ہیں بیک کراسنگ تو کرتے ہیں آپ, لیکن یہ ہے کہ پیرنٹس اس میں آپ چینج کرتے رہتے ہیں, اور ایک پول سا آپ نے پول بنایا.
سر آپ کی عظمت کو سلام, آپ نے اتنی بڑی کراپ کو لے لی, کیونکہ 25 ایئرس اس میں لگتے ہیں.
نہیں, میں نے تو کچھ نہیں کیا ابھی تک.
تو سر جو باقی اسٹوڈنٹس ہیں پوسٹ ڈاک کے ہمارے پی جی اسٹوڈنٹس, اس لحاظ سے تھوڑا سا یہ ڈسکربنگ لگتا ہے کہ اس میں ٹائم کافی لگتا ہے. تو اس میں کیا ہے اٹریکشن, اس میں جو آپ نے جونیئر اسٹیج بتائی ہے تو اس کو کم کرنے کے لیے کس پر ٹیکنالوجی یا کوئی نئی ٹولز ہیں سر جو ہم یوز کر سکتے ہیں؟
صحیح. پہلے تو تھوڑا سا میں کلیریفائی کر دوں, وہ کلیریفائی ایسے کر دوں کیونکہ کورنل میں ہم ایپلز جو ہے اس پہ تقریباً کتنے 10 یا 15 فیکلٹی ہوں گے جو ایپل پہ ریسرچ کرتے ہیں, اور اس کے ڈفرینٹ اسپیکٹ پہ کرتے ہیں. یعنی میں ہی میرا ایک چھوٹا سا وہ ہے, وہ فائر بلائٹ اور ایپل اسکیب پہ چھوٹا سا کنٹریبیوشن. ٹھیک, تو کورنل میں ایپل بریڈر بھی ہے جو سائن بریڈنگ کرتی ہے, سوزن براؤن, پروفیسر سوزن براؤن. کورنل میں ہے ہارٹیکلچر پروفیسر ہے, ہارٹیکلچر پہ کام کرتا ہے, پروننگ اور ٹریننگ ٹریز کی جو ہے, یعنی وہ سب کے میں نام نہیں. اسی طرح جو روٹ سٹاک ہے اس کے الگ ٹریٹس ہیں, اس کی الگ وے ہے اس کو بریڈ کرنے کی, اس کے لیے ایک الگ سا گروپ ہے. میں بھی اس گروپ میں ہوں, کو بریڈر یا کو انوینٹر. لیکن یہ ہے کہ اس میں بھی 2-3-4 لوگ ہیں, تو ہم سب لوگ ایز اے ٹیم جو ہے کام کرتے ہیں. تو میں پری بریڈر میں اپنے آپ کو کہوں گا, ٹریننگ سے تو میں بریڈر ہوں لیکن میں پری بریڈر اپنے آپ کو کہوں گا. میں وہ فروٹ کوالٹی والا جو ہے نا وہ کافی کمپلیکس ہے, اس میں نہیں پڑتا وہ, اور وہ سب سے امپورٹنٹ ہے کیونکہ کنزیومر کوالٹی کے بغیر تو نہیں کرتے. تو سوری آپ کا ابھی میں کوشچن ہی بول گیا, وہ جو ایکچوئل کوشچن.
جو بایو فورٹیفیکیشن کا ہے پروجیکٹ, اس میں آپ نے کوئی کام اس میں کیا اور ایپلز کے لحاظ سے؟
نہیں, بایو فورٹیفکیشن میں نے تو نہیں کیا.
سر میں آزاد کشمیر کی ٹوپوگرافی کو بڑی اچھی طرح جانتا ہوں. آپ نے تو میرے بات میں وہاں پہ سروس کی ہے, تو آپ نے سر آزاد کشمیر کے لیے گورنمنٹ کے لیے کیا کام کیا ہے, کیونکہ وہاں پہ کنڈیشن کافی زیادہ ہے.
میں پوری دنیا کے لیے کر رہا ہوں, آزاد کشمیر تو آپ مجھے جذباتی کر رہے ہیں, نیشنلسٹ بنا رہے ہیں. نہیں, اس میں یہ ہے کہ جو ہم ایکیڈیمک ریسرچ اسپیشلی جو ینگ لوگ بیٹھے ہوئے ہیں, ہمیں گلوبلی سوچنا چاہیے. اور یہ ہے کہ آزاد کشمیر میں جو یعنی اس میں نے جو انٹرنیشنل ڈیولپمنٹ میں کام کیا ہے, انٹرنیشنل ایگ میں, اس میں ایک چیز آپ کو شاید آپ اسے ایگری کریں یا ڈس ایگری کریں, لیکن میں نے دیکھا ہے کہ جو بھی ملک ایگریکلچر میں اسٹرانگ ہیں وہ اسٹرانگ ہیں, ان کا کمرشل ایگریکلچر اسٹرانگ ہے. وہ جو چھوٹی ٹرینز پہ چھوٹا فیلڈ ہے جس کا ہولڈنگز ہیں, اسمال ہولڈرز جو ہیں, وہاں پہ غربت ہی دیکھی ہے میں نے. اور اس کی وجہ یہ ہے کہ اگر جیسے آزاد کشمیر کی میں بات کر رہا ہوں یا کہیں بھی بلوچستان میں کریں, NWFP کے کسی بھی ملک میں کریں, تو جب آپ 10 بندے یا 5 بندے ایک ایکڑ میں لگے ہوئے ہیں اور اس سے 2 من یا 10 من سیب نکلتے ہیں تو اس سے کیا بنتا ہے؟ اس سے نہ آپ کی ایجوکیشن ہو سکتی ہے، نہ آپ کی ہیلتھ ہو سکتی ہے، نہ تو یو ایس میں جو ہے وہ ایک گرور ہوگا، اس کے پاس ایک تو ہابی ہے میرے طرح کے، وہ تھوڑے تھوڑے پودے لگے ہوتے ہیں، وہ تو ایسے شوق ہے جو کمرشل اکنامکلی کرتے ہیں بزنس، وہ ایز اے بزنس رن کرتے ہیں، ان کی وہ ہابی نہیں ہوتی۔ وہاں پہ میں نے دیکھا ہے جو بزنس مین ہے جس کا ایپل کا آرچرڈ ہے، اس کا 100 ایکڑ ہوگا، 200 ایکڑ ہوگا، 500 ہوگا، 1000 ہوگا۔ اسی طرح ٹرکی میں بھی میں نے دیکھا وہاں پہ جو لوگ ایپل سے پیسے بنا رہے ہیں وہ ایز اے بزنس اس کو رن کرتے ہیں، اور وہ ان کے ڈیسیژنس بھی اکنامک ہوتے ہیں۔ اگر ایک ایپل گروور میں نے دیکھا ہے، اس نے ایک ورائٹی لگائی ہے اور وہ ورائٹی 50 ایکڑ پہ لگی ہے اور ابھی ینگ ہی ہے، لیکن اس نے دیکھا ایک نئی ورائٹی آئی ہے، اس میں میری کمپیریٹیو ایڈوانٹیج ہے، اس نے اس کو اکھاڑ کے پھینک دینا ہے اور وہ دوسری لگا دینی ہے۔ اس نے اکنامک ڈیسیژن کرنا ہے کہ اس میں 5 سال کے بعد میرا کیا فائدہ ہوگا۔ تو آزاد کشمیر پاکستان میں بہت سی جگہوں پہ یہ چیزیں ابھی تک نہیں ہیں۔ وہ جو میکنائزیشن ہے اس کی وجہ سے جو ہے ایک ہی بندہ وہ 50-100 ایکڑ جو ہے نا اس کو رن کر رہا ہوتا ہے، تو ہمارے پورا گھر پورا گاؤں جو ہے لگا ہوتا ہے تو آزاد کشمیر اس اسکیل میں بہت چھوٹا ہے۔ ابھی وہ جو انڈیا والی سائڈ پہ ہے وہاں پہ بھی ہم تھوڑا وہ کرتے ہیں ایگریکلچر میں، یا ایپلز کے ساتھ بھی، یا پوٹیٹوز میں بھی پہلے تھا، تو وہاں پہ بھی ہماچل میں یا ایٹ لیسٹ کشمیر میں، جو اس سائڈ پہ کشمیر ہے شملہ میں، تو انہوں نے شروع کیے ہائی ڈینسٹی آرچرڈس۔ تو یہاں پہ آپ نے ایک فوٹو دیکھا ہوگا، کیونکہ ہم آپ پنجاب میں تو ایپلز نہیں ہوتے، تو آپ پتہ نہیں کس طرح کا آپ کا جب ایپل کا آپ سوچتے ہیں تو کیسا آپ کو دکھتا ہے کہ ایک بڑا سا ٹری۔ یو ایس میں جو ایپل آرچرڈ ہے اس میں آپ جائیں تو آپ کو، میرے پاس اچھا فوٹو نہیں ہے یہاں پہ، پلانٹ ٹو پلانٹ ڈسٹینس جو ایپل کا ہے وہ 3 فٹ کا ہے، 3 فٹ، مطلب ایک پودا یہ ہے دوسرا یہ ہے، تو وہ ایسے لگتا ہے جیسے ٹماٹر لگے ہوئے ہیں۔ اس کو ہائی ڈینسٹی آرچرڈ سسٹمز بولتے ہیں۔ اس میں یہ ہے کہ جیسے یہ فروٹ نظر آ رہا ہے آپ کو، اس پہ لائٹ جو بہت ہی یونیفارم پڑتی ہے، اس سے کوالٹی یونیفارم آتی ہے۔ اس کے بعد اس کو سپرے کرنا ایزی ہے، اس کو پروننگ کرنا ایزی ہے، اس کو ہارویسٹ کرنا ایزی ہے، میکنائزڈ ہے۔ رو ٹو رو ڈسٹینس جو ہے وہ 10-15 فیٹ ہے، ٹریکٹر اس کو ایزیلی وہ کر لیتا ہے۔ تو وہ اگر آپ یہاں پہ کریں تو انڈیا میں انہوں نے وہ شروع کیا ہے، انہوں نے، اور اس کے لیے جو پاسبلٹی ہے وہ روٹ سٹاک، اس کے لیے پاسبلٹی ہے، جو روٹ سٹاکس ہیں اس میں ڈوارفنگ جین ہے جو روٹ سٹاکس کو ڈوارف رکھتا ہے، تو وہ ٹری بڑا نہیں ہوتا بہت بڑا، اس میں جین ہے ڈوارفنگ جین، تو وہ جین جو ہے وہ ٹری کو ایک سرٹن ہائٹ ہے اس سے اوپر نہیں جانے دیتے۔ تو
ورٹیکل اس کو جو ہے نا وہ زیادہ نہیں کر سکتے، یعنی آپ جو اس کا سائن ہے روٹ اسٹاک تو روٹ کو کرتا ہے نا، تو جو اس کا سائن ہے اوپر والا وہ اس کی یعنی پروننگ اور ٹریننگ اس طریقے سے کرتے ہیں کہ اس کو ورٹیکل لے کے جاتے ہیں۔
بالکل ایسے بھی ہے، اور پروننگ ٹریننگ سسٹم بھی ڈفرینٹ ہے، ایون نیوزی لینڈ آسٹریلیا میں بھی اور یو ایس میں بھی ڈفرینٹ جگہوں پہ ڈفرینٹ ٹریننگ سسٹم ہے، وہ لائٹ کی وجہ سے یا مشینری کی وجہ سے۔ اس جیسے میں کہہ رہا تھا کہ ہمارا ایک پروفیسر ہے ٹیرنس رابنسن، اس کا ریسرچ ہی پروننگ ٹریننگ میتھڈس پہ ہے، تو بہت سارے اس میں وی شیپ ہے، اس میں سینٹرل لیڈر، بہت سے ٹریننگ پروننگ سسٹم بھی ہیں۔ سوری،
Hi, good afternoon.
اچھا،
Thank you so much for the insightful lecture. And this story was very insightful about experiences from a remote village in Kashmir to the Ivy League college. I think every student dreams of going to an Ivy League college here. And so, as a student, I would like to ask, when you receive an application from a student, what is something which uniquely stands out in that student for you? And while you were working on apples and potatoes, and then you went for a postdoc in medicinal plants, how did you relate your statement of purpose from potatoes and apples research to medicinal plants, and how we can relate our research from cotton or potatoes to apples or something if we are applying for another research project.
So it's a very good question, and I think if I just open my email now, you will see maybe 10, 15, 20 emails about admissions in PhD or postdoc or and so on. And I think the reason I'm saying this is almost everybody in the US or Europe or different places, they get so many emails from students, and it's impossible to also read so many emails and respond to so many emails and also having so many positions open. Yeah. So there has to be some method of selection, screening. Yeah. So I think one thing is, I mean, now there are also many factors. So for example, sometimes professors, they don't respond because they don't have position. They are retired. They are not recruiting anybody. There are so many reasons. But also if somebody is recruiting and then they are not responding to you, I think one thing could be if this is not a genuine email. If it is not real, from a human, but also a genuine, like, for example, you asked me this question. You asked me this question genuinely, yeah? So other person can tell that this is a real email or it's just a superficial. And I think if you want to get response from somebody, you have to write it genuinely. Because otherwise, all other 14, 15, usually those 14, 15 are just superficial, generic emails. And now, after seeing so many emails, all of us, we have learned how to ignore or how to screen that this is genuine and this is not genuine. Yeah. So I think what I'm saying is, like anybody, any professor or anybody who's recruiting somebody, your professors here, if they're recruiting somebody, or let's say you are running a shop, yeah, you want to have somebody, you want to recruit somebody for your shop who can boost your business. And the only way you can tell is if the person is genuine and has done some kind of homework before approaching you. Yeah. So I think if you write an email to me and you say you are interested in Cornell or US, very broadly, I don't care about Cornell, I don't care about US. I think when I interview students who are applying in my program, I say my main motivation is asking them why they want to work with me. And I try to ask different questions like, okay, first they will say, okay, I want to be at Cornell because Cornell is the best school. Okay, I am not Cornell. I am running a research program on apples and genetics of apples. I mean, of course, I am from Cornell, but Cornell hired me to work on that aspect. So anybody who's passionate about genetics of apples, doesn't matter what is their previous background, if they because it can click in your mind at any point of your life that you're really interested in this area. And if you can show that, then I think I will be very happy to have you. I think one thing, we in academics, we are not there to make money. We want to have our legacy. Legacy means I want to see this area of research going forward. And this area of research will go forward if there is somebody really passionate about this area. I want to see I retire and you continue this. So this is, I don't want to waste my time of life on something that doesn't push this area of research forward. I think that's, so if you can show that you are the one who is really genuinely interested in this area and you can push it forward, I think then I will be stupid not to take you. I mean, this is, everybody is looking for those people who are really going to push that area forward. And it is sometimes you can we are people, we can make misjudgments, we can recruit somebody who is misappropriate, but that is always the case. I think when I went to Germany and Switzerland and all these things I told you, I used to hear that, oh, there's a discrimination, there is this and that. My full name is Muhammad Awais Khan. I never see any discrimination. Nobody discriminated me. I mean, I give example to students sometimes that there is my brother and there are other people in the village, and I have again a shop, and my brother is not very, good in running business, is not reliable. So I will be stupid to give that job to my brother. I will have somebody who is really appropriate and is good, hardworking. So I think this is everybody is looking for that. So I will, and to have those specific emails, there is no magic. The only magic is go and look at their web pages. I mean, like I am here bragging about my research. I am 30 hours tired. Yeah? I still want to talk about my research because this is my life. Yeah. So the same thing is, if you read my papers or the website and then write to me, I will be so excited. Oh, like a musician, they want to always, sing to you. So I will be happy. So I think this is what, if you can convey to any professor genuine interest after looking at their pages, because if you don't do that and you write to them, they can really spot that this is just a superficial email. I mean, the same thing I can do to you, and you can see that this he doesn't know. I also say, for example, iPhone. So if you are really into phones, if I ask you about the camera model and pixel and everything, you will tell me everything about the phone. The same thing is, if you are passionate about apples, you will learn about it. So I think develop that deep insight and then write to them. But still, if they don't respond, I think there are many other reasons as well. As I said, maybe they are closing down their lab, they are moving to another position, they don't have funding. There could be many reasons.
And recently, like Trump has stripped off funding for most of the labs in USA. So does that affect, future research projects in your lab as well?
I mean, that has impact for all of us. But those things are beyond our control. Yeah. So I think we have to find our way and navigate our way from that. Yeah. I mean, we cannot change Trump. So, Cornell University is a very big university, but we also have big economic, financial impact. So, I mean, yeah, are you recording what I'm saying?
Thank you. We can have one more question from Dr. Osman Arif.
Thank you so much for a very interesting talk, because generally we don't talk much about apples. I just want to know about your take on GMO apples. I believe GMO apples, genetically modified apples, I believe they are not accepted as yet. So what is the prospect of GMO apples in future? What is your take on it? And related to that, given the advancement in genome editing tools, do you think that we can use some sort of tissue-specific regulations so that it doesn't affect the fruit, but it can improve the rest of the plant architecture?
Okay, so about the GMO, I think there are RNAi silenced apples in the US. You can find them, Arctic apples. These are non-browning apples. PPO enzyme is silenced, and you have non-browning. You can find in different stores in the US. So I think maybe it's not correct to say that there are no GMO apples. In Europe, there are regulations which are different, but in the US, there are this RNAi silenced apples. You can find them. So they silence this enzyme in Gala, and they have Arctic Gala, they have Arctic Granny Smith, and different Arctic apples. And what is my take on GMO? I mean, I'm a scientist, and I don't see any issue with GMOs. I think this is the technology which is most regulated than any other technology. For example, chemicals and pesticides and those. I mean, they're also regulated, but I think it's a very deeply vetted. So every single gene, we know usually the function and how the impact will be. But also, I work with breeding. Yeah, I work with natural diversity, and I also think the first thing for me to look is the natural diversity. But I also don't discount the GMOs. I mean, the same way. But I think conceptually, if we look all the time, there are crosses, there are mutations happening, and we have never thought about, okay, this is are the evolution and all those processes and the mutations. I mean, in apples, this is happening all the time. For example, you have Gala, and you have like maybe 30 point mutations or sports of Gala. We call them point mutations, but I don't know what other changes have happened to them. So, but they are considered non-GMO, and then you have one gene inserted where you know the function and it's well characterized, and you say it's GMO. So, anyhow, as a scientist, I don't think there's any issue, but it's the regulation of the countries, and we respect the regulations of the countries. But I think conceptually, I don't think there's an issue.
Yeah, because I think when you are screening so many genotypes or cultivars, some will inherit, natural resilience against biotic and abiotic. So we can leverage that weight, to transfer from stress tolerant species to the stress sensitive ones. So that's why I was asking about genome editing tools and functional validation of the gene. That is the way to go in future.
Yeah. Yeah. Okay, that's it.